More than 25 years ago, my family experienced a tragedy that many of you have unfortunately shared. My father was struck by a mystifying set of rapidly progressing, terrifying symptoms. He was eventually diagnosed with Creutzfeldt-Jakob disease, and within a few months, he passed away. This experience was so far removed from my sense of reality that I literally felt like I had stepped into a twilight zone. As my family and I grieved my father's loss, we were overwhelmed by what he had experienced and dismayed by how little was known, how little could be done for him, and how alone we felt. Our family was determined to make an impact on the CJD trajectory. Dr. Joseph R. Berger, my father's physician, connected us with Cecile Sardo, whose husband he had previously diagnosed with CJD, and who, together with her son, Joseph, also yearned to do something in response to this terrible disease. Together, the three of us created the CJD Foundation 25 years ago in Miami, Florida, and began our journey to raise public awareness of CJD and related prion diseases, educate medical professionals and the public, promote research, and support families and friends of CJD patients. In 2000, we hosted our first CJD Foundation conference. In these 25 years of the Foundation's evolution, my initial overwhelming feelings of grief, despair, and isolation have been replaced by an overpowering sense of hope, purpose, and most of all, community, of which the Foundation has been both a catalyst and beneficiary. Our unique CJD Foundation community, made up of the people whose lives have been impacted by prion diseases, as well as the scientists, doctors, and healthcare professionals who work on and with these diseases, have made possible all of our inspiring accomplishments. Our amazing journey together makes me confident that we will be moving towards an effective therapy or cure in the years ahead.
A note from Florence Kranitz. After losing a dear friend to this terrible disease, a small group of friends in Akron, Ohio, vowed to make a difference in his honor. In 2002, after forming a task force and working beside the founders of the CJD Foundation, they were invited to take over the foundation leadership and moved the office from Miami, Florida, to Akron. The elected officers, Florence Kranitz, President, Mark Goldfarb, Chairman of the Board, Nick George, Vice Chairman, Ruthie George, Treasurer, and Anna Bitro, Fundraising Vice President, were fortunate to be carefully guided by Dr. Pierluigi Gambetti, Director of the National Prion Disease Pathology Surveillance Center, who agreed to become the Foundation's medical director. With an extremely dedicated board of directors and a generous grant from the Centers for Disease Control, the CJD Foundation was able to carry out its ever-expanding mission. In reflecting back over the years of this group's involvement, Florence indicated she is proudest of the establishment of the helpline, initiating the annual family conference, helping to form the CJD International Support Alliance, and the introduction of family-funded research grants.
A note from Dr. Pierluigi Gambetti. I became the director of the National Prion Disease Pathology Surveillance Center, located in Cleveland, Ohio, in 1997. And subsequently, when it was moved to Akron, Ohio in 2001, the CJD Foundation asked me to become its medical director. I retired from both in 2015. During the 14 years of my close interaction with the Foundation, I had the great fortune and the very rewarding experience of working closely with the Foundation President, Florence Kranitz, and the other leaders, especially Ruthie George and Lori Nussbaum, as well as the advisory board. It was exciting to see the Foundation and the Surveillance Center grow together, a growth that continues under the current presidency of Debbie Yobbs and the directorships first of Yuri Safar and now Brian Appleby. However, when I think back to those remarkable years, it is the memories of the family meetings that stand out. I felt the incredible, enduring pain that these terrible diseases can cause to the affected families. But I also witnessed countless examples of courage and strength to overcome their losses and find new meanings. To me, a pathologist who mostly sees diseases through the microscope, this was a moral lesson and an inspiration. It provided me with a different perspective of my work and what I could contribute through it. I treasure these interactions and the relationships that ensued, and I miss them. I wish the CJD Foundation many more happy 25th anniversaries, and I salute you, the family members, with great affection and admiration.
When my husband was diagnosed with the prion disease 12 years ago, I was incredibly grateful to be able to contact the CJD Foundation for information, support, and referrals. Today, I'm honored to lead the CJD Foundation as president. Over the past several years, we've been pleased to be able to enhance and build on the Foundation's outstanding initiatives, such as medical education, helpline, advocacy, and family workshops. We've added programs like teleconference support groups and expert lectures, bringing families across the country access to information and updates from clinicians and scientists in the prion disease field. Strides for CJD, launched in 2015, has not only been a successful fundraiser, but also has served as a source of moral support for families and has raised awareness of CJD around the country. And most importantly, we have continued to expand the size of the CJD Foundation's research grant program. From 2015 through 2018 alone, we have awarded close to $1 million in research grants, thanks to generous donations from families and supporters. As we celebrate the 25th anniversary of the CJD Foundation, we look forward to continuing to support families affected by CJD, educate medical professionals, and push forward on our goal of finding a treatment for prion disease.
I was fortunate to be introduced to the CJD Foundation by Dr. Paul Brown early in my career. My initial query to Florence Kranitz was whether or not the field needed more clinicians and clinical researchers. I think you can guess how she answered. For over a decade, I've had the privilege to work closely with the Foundation. Despite being a small group of people, I've seen it grow and innovate much more quickly than other patient advocacy groups. The reason for this is simple: passion. I've met some of the most amazing and hardworking people through the Foundation that all share the same passion in assisting families affected by this terrible illness. My hope is that we will have a treatment for prion disease over the next 25 years, and I have no doubt that the CJD Foundation will play a large role in making this happen.
Good morning, everyone. It's a real pleasure to be here. This is my third time at this meeting, and it's a crucially important meeting for us for obvious reasons.
I must say, every time I come to this conference, I find it very educational and moving and motivational to me and to people in my lab as well, to hear all of the real stories from those of you who really have to deal with these diseases. And it does a tremendous amount to stoke our fires in the research world to solve some of the problems that are ahead of you and all of us together.
The subject was to use human mini-brains as a new platform to identify and validate pharmaceutical compounds for the treatment of CJD. So a very ambitious program, as you see. And I must say that it's the first time that we are asking for a grant from the patients, and it was very specific. And I told my students and all the group that it was not neutral money there. It's not a neutral grant. Behind this you have pain and you have a lot of hope. And we need to be at the level of this expectation. So my students and my group had high pressure about it, and you will see that it was a positive pressure.
As they say, this isn't rocket science, but really what makes this an epic setting idea is the fact that it's not just PRPC turning to PRPSC and that's the end of the process. PRPSC then acquires the ability to convert PRPC, the normal cellular protein, into more of the disease-associated form. So a good guy and a bad guy entered the scene, and two bad guys leave. So that's basically the prion conversion in a nutshell.
So I'm going to talk about penguins. They're nice.
This shows that we were able to introduce good anti-PRP, CWD titers in the saliva of the deer, which is important for protection, and also in the feces. This was not a popular job, extracting IgA from the feces in the lab.
If you had asked me several years ago, before I knew that there was anything in Sonia's family, what would you do if you or someone you love had a fatal genetic mutation? I would say I have no idea. Probably just cry a lot. But it was only after we got the test results that suddenly we found ourselves doing things we had never imagined.
Some of the cells are sensitive and actually express PRPSC when they're exposed to prions, while others are resistant. So it's a really great way to say, okay, and ask the question, well, what makes those cells sensitive and what makes those cells resistant?
My lab has decided to actually focus on something that we know very well, which is the cellular form of the prion protein. For this protein, there are several advantages of deciding to go with a drug screening against the cellular prion protein.
We have therefore artificially created a human antibody called PRN100 that binds tightly to the normal prion protein. This can be manufactured in large quantities for use as a drug given by injection. The aim is for the antibody to bind to the normal prion proteins on the cell's surface and block their interaction with the rogue prions. This prevents the prions growing further and stops the chain reaction. We know that the brain can slowly get rid of the remaining prion chains by its own natural mechanisms once they have been prevented from growing.
We're all brought here by suffering and by having lived through an incredibly difficult experience. I know firsthand that can really, there's a temptation to be angry at some of the moving parts in the system that seem to be in the way of therapeutics. There's a temptation to be angry at physicians or at researchers or at regulators or at any of the sort of players in this complex system that we have for bringing drugs to patients. But if there's one thing I can say as a person who's a little bit of a bridge between two of these worlds, it's please try to open your hearts as much as you can to the ways in which we need all of these people and the ways in which it really is a miracle that they come together time and time again to produce drugs that treat disease. And, we have drugs on the shelf today, and I hope that soon, someday, we'll have one for our disease too. So, thanks very much.